Okay, madam. I'm Dr. Aisha from Government First Grade College, Tumkuru. It's in Karnataka. So my topic of today's presentation is biogenic Leucas aspera fueled cadmium sulfide nanoparticles. Its synthesis, characterization, investigation of catalytic activity. So what we have done in our work is like we have used Leucas aspera leaves. These are known as tumbe plant locally. Using this as a fuel, we have prepared the cadmium sulfide nanoparticles. Later on, as usual, we have characterized them. And after the characterization, we have checked or investigated it for two different application parts. One is for the synthesis of an organic compound, a series of organic compounds, etc., tetrahydrobenzopyranes by a multi-component reaction, and finally the corrosion inhibition. So if you all can see on the screen, this is the plant that we have used, the leaves of this plant. This is a locally available medicinal plant having a very good activity as it works as medicine for colds, coughs, and various other things. It's a very common these leaves and the extract, the fresh leaves we have taken, the extract has been taken. It's approximately like 10 grams in 100 ml. We boiled, and the extract has been taken along with sodium sulfide and cadmium sulfide mixture. And this mixture was homogenized and taken in silica crucibles. And under muffle, it was kept for 2 hours and calcined at 700 degrees to get a white powder, which was desiccated, and for further characterization, it was used. Characterization was carried out with the usual techniques, spectroscopic techniques. One is IR and the other one is XRD. So these two confirm the that these is the obtained nanoparticles were in good purity. And what we have done is we also have done the same analysis at 20 and 50 micrometer resolution. We can see that they are agglomerated and clustered. The elemental analysis proves the amount of sulfur and cadmium at 44 and 55% approximately. In the further application part, we evaluated these things for synthesis of an multi-component reaction using aldehydes, dimidone, and malononitrile to prepare a series of benzopyranes, and we have used different aldehydes here. They were obtained in very good yield. The reaction was carried out under reflux in water at 80 degrees, and we used only since they are heterogeneous catalysts. It was easy to remove them, and the product was dissolved, and the purity was checked by the TLC, and we found that they were very good. And here I have just presented one of the IR of one of the molecules that we have prepared. And further, we decided to check the corrosion inhibition efficiency of these cadmium sulfide nanoparticles. So what we did was like we sourced out the mild steel of grade IS 513, and we made the coupons of 1 by 1 centimeter, and we carried out the corrosion inhibition. We tried to simulate the the seawater type of environment, corrosive environment for that. For that reason, we have taken 5 grams, 5% NaCl, by dissolving 5 grams of sodium chloride in 100 ml of distilled water. And the coupons were immersed in this corrosive media for 24, 48, and 74 2 hours. And after every one cycle, we used to take it out, take their weight. Previously, weight coupons were used, and after the 1 day or like 2 days or something, we used to take them out and dry them, take their weight. And this was that weight loss studies from which we found that these exhibit good corrosion inhibition at our lab conditions. So this was the another application part. So one was the use of these for the synthesis of hydrobendoperones. The other one was corrosion inhibition. This was a very basic research which we were able to carry out at our simple setup. So I would like to conclude that we have synthesized cadmium sulfide nanoparticles using Leucas aspera plant leaf extract, and we have characterized this, and they show good inhibition as well as catalytic performance. Thank you.
Today I'm presenting my work on treatment of college canteen sullage using microcosm phytorid system. These are the contents of my presentation.
Okay.
So coming to the introduction part, as we all know, water is becoming a rare resource of the world now, and almost 1 in 3 Indians might face acute water scarcity in the near future, as per the International Water Management Institute. And also coming to my work, wherein I'm concentrating on the treatment of sullage. Sullage in domestic sewage, it accounts to 80%, in which 44% of it is generated from the kitchen outlet. And if an appropriate method is being used for the treatment of this sullage, definitely it will serve as a significant factor to overcome the water crisis for non-potable use. And among the various treatment processes, the phytorid wetland proves to be the economic and efficient system to reduce the pollutants in the sullage. So here, the objectives of the study include characterization of sullage sample from the college canteen. We had collected the college canteen sullage itself for the treatment, and the selection of suitable types of wetland plants for the treatment of sullage, and a comparative study to check the effectiveness of the plants in treating the canteen sullage. So the materials and methods that were being included, the first one, that is the sullage collection. It was being collected in a PVC container of 20-liter capacity, and it was being sedimented for 24 hours. And the oil that was being collected on the surface of the sullage, it was being skimmed off, and the sludge, as sedimentation was being the sullage was being sedimented, the sediment was also being excluded from the treatment. And then the next is the phytorid bed. It constituted of 5 microcosm phytorid systems, small scale phytorid systems, wherein a plastic containers were being used of 35 centimeter length by 23 centimeter breadth, and height was 17 centimeter. And these containers were filled with 3 layers of substrate, starting from the bottom to the top, that is gravel, sand, and garden soil. And then 5 different plants were being planted in these 5 containers. To start with, the first one, that is Canna indica, which was being coded as P1, Moses in the cradle P2, and the spider plant as P3, and the golden duranta plant as P4, and beach spider lily P5. These were planted in 5 different containers. And the treated sullage effluent was tested at an interval of 7 days, that is after 7th day, 14th day, and 21st day, from the control as well as the 5 microcosm phytorid systems. The control here was not being planted, but the substrate was the same here, with the 3 layers of gravel, sand, and garden soil. And the parameters such as pH, electrical conductivity, chloride, sulfates, phosphates, total dissolved solids, BOD and COD were being analyzed. And the treated sullage effluent characteristics from the phytorid systems with different plants were evaluated. So this is the basic setup for the materials that were being used here. That is the gravel, sand, and the garden soil. And in the figure D, you can see the layering of these substrates that were being used. So these are the 5 plants that were being used, and this is the complete setup, wherein a 20-liter can was being used, which was being filled with the sullage collected from the canteen, kitchen, kitchen outlet. And here the container in which we stored the sullage, it also served as the sedimentation tank. And from there, it was being sent to the phytorid bed through the connections which are being used for the drip irrigation in that way. And the treated effluent was collected in the collection unit, and that was being analyzed. So now these are the variations in the physicochemical parameters for the 3 weeks for which it was being tested, wherein the raw sullage was also being tested, and the control also, the effluent coming from the control was being tested, and from the 5 different containers consisting of the 5 different plants was also being tested. And here the pH, it varied, it was acidic in the beginning, that is in the raw sullage, which was 4.6, but whereas in the containers with the plants, P3 showed 7.14 pH, which raised to the alkaline range, and then P2 plant it showed 7.56, and electrical conductivity from 1600 microsiemens per centimeter, it was reduced to 1436 for P5, and for P4 it was 1579. And then the chloride level, it reduced from 387.5 to 76.5 for P5 plant, and sulfate, it reduced from 136.25 to 87.65 milligram per liter. And the phosphates, it reduced from 13.63 to 3.55 for P3 plant. And then the TDS also reduced from 1150 to 876 milligram per liter for P5 plant. And BOD reduced from 405 to 54, which was observed for P4 plant, and COD from 535 to 145 for the P3 plant. Similarly, for week 2 also it was being tested, and these were the values where significantly there was some reduction being seen for the different parameters that were being tested. And for week 3 also it was being tested. And here we could see that the pH from 4.89, it was being like raised to 7.32 for P1 plant, and 7.89 was the pH observed for P5 plant. And electrical conductivity from 1575 microsiemens per centimeter, it was reduced to 1324 for P5 plant. And then chlorides from 345.43 milligram per liter to 42.54 milligram per liter was being observed for P5 plant. And sulfates also and the other parameters were being reduced like this, and the same the reduction efficiency we can see here with respect to chloride, sulfates, phosphates, TDS, BOD, and COD for the 3 different weeks which were being studied. And then from the results, what were being obtained from the analysis, it was found that the spider plant, which is P3, was efficient enough in reducing sulfates by 38.69% and TDS by 30.17% and COD by 75.05%. And the P4 plant, that is Golden Duranta, it exhibited higher reduction with respect to BOD, that is 88.94%. And then the beach spider lily, the P5 plant, it showed higher reduction for chlorides, that is 87.68%, and phosphates 79.43%. So from this study, the conclusions that were being drawn are the laboratory study was done on a small scale with the constructed wetlands to treat the college canteen sullage, and the experiment was done using 5 different species available on the college campus. And it was conducted for a period of 3 weeks, and in this study the P3, it showed an efficiency for reducing sulfates, TDS, and COD. And the Golden Duranta plant, it exhibited higher reduction with respect to BOD, whereas the beach spider lily plant showed higher reduction for chlorides and phosphates. So from the obtained results of the study, it can be concluded that these 3 plants in combination may be used for effectively treating the canteen sullage, and which can be brought down to the standard level so that it can be used for the purpose of gardening and flushing on the campus. So these are the references that have been referred. That's it.
Myself, Dr. Mahesh Bhat, Assistant Professor working in the Chemistry Department, Pune College, Udupi. It is in Karnataka State. My research topic is photocatalytic degradation of aqueous dyes by titanium epitote starch nanocomposites. In this study, we have synthesized the titanium-based nanocomposite material and analyzed it for the SEM and DLS for particle size distributions, and utilized it for the photocatalytic oxidation of the three dyes, namely food color yellow, electron black tea, and methylene blue dye solutions. Among the semiconducting metal oxides investigated, titanium dioxide, zinc-based cerium, bismuth, and tungsten-based nanocomposite or the nanomaterials are getting the more attention because of their photocatalytic activity. The synthesis of the metal oxides nanoparticles have higher efficiency and low-cost material for the functionalized materials, and exhibits the good hydrophobic and magnetic properties also because of the enhanced surface area. Nanostructure materials have been employed as efficient photocatalysts or as hybrids or in the combined form or in a doped with other substances to aid in the degradations of the various organic pollutants. Titanium dioxide nanocomposites has high chemical stability, inexpensive, and non-toxic. Titanium dioxide polyvinyl alcohol nanocomposites was evaluated for the electrical properties and as applications as anti-reflection coatings in the solar cells. Thin film of the polyvinyl nanocomposites was decreased the band gap from the 3.94 electron volt to 3.04 electron volt when significantly titanium dioxide was doped. Water pollutants encompasses with a wide range of the compounds, including pesticides, dyes, pharmaceuticals, and the polycyclic aromatic hydrocarbons. The photocatalytic activity of titanium dioxide is significantly influenced by the properties such as crystalline phase, crystalline size, and the specific surface area. Titanium dioxide photocatalysts with a large surface area exhibit high photocatalytic activity. Additionally, their small size causes them to the agglomerates into the larger particles, reducing the surface energy and catalytic performance. To address this issues, various methods such as desensitization, doping, coupling, and different methods have been studied, and the different nanocomposite material has been synthesized in the literature. In our research work, the preparations of the titanium-based nanocomposites are synthesized by using perchloride of about 2.027 gram and 2.0 gram of the titanium dioxide, and 2.5 gram of the starch is added in a reaction mixture, and 3 gram of the sodium hydroxide solution was added and stirred for the 30 minutes, and filtered and collected and dried in a mobile furnace about 2 hours at 100 degrees Celsius. And after drying, the powder was sieved that all the particles to be utilized for the uniform size. Dye solutions was prepared by taking the food color yellow, methylene blue, and erythrom black tea at 50 ppm concentrations. The study involved in exploring the various parameters such as contact time and initial concentration of the solution for the effective removal of the color in a batch experiment by photocatalytic actions of the synthesized nanocomposites. Each experiment utilized 50 mg of the synthesized nanocomposites, allowing for the systematic investigation into the degradation process under the different conditions. And the results, this table shows that the percentage of removal of the food color yellow dye with respect to the contact time in 320 minutes, there is a 49.24% of the photocatalytic oxidations of the dye has been taken place. In the case of the erythrom black tea dye solutions, here the results shows that there is a moderate removal of the color in a dye solution of about 16.54% removal of the dye. In the case of the methylene blue, there is a 22.48% of the removal of the color taken place. This graph shows that the comparative study of three dyes, that is methylene blue, erythrom black tea, and the food color yellow. This shows that the efficient removal of food color yellow dye taken place by the synthesized nanocomposite materials. This image shows that scanning electron microscopic image, and particle size distribution also determined by the DLS method, and it shows that around 100 nanometer of the particle size. The present study synthesized the nanocomposite material from the titanium-based biosulfate and the starch by the solution phase method. Synthesized composite exhibited moderate activity on the particular yellow, erythrum black tea, and methylene blue dyes in aqueous solutions. In the literature, many of titanium-based nanocomposites materials were synthesized and utilized for the various study. Here we reported for the dye degradations, that is photocatalytic oxidation. If the studies continue, it may emerge as one of the excellent nanocomposite materials. The study involves in the synthesis of the nanocomposites materials and removal of the dyes from the aqueous batch solutions. Here we have carried out here three dye colors, that is erythrum black tea, methylene blue dyes, and the porticolor yellow. There is a 49.2% removal of the dye in the porticolor yellow taken place by the synthesized nanocomposite materials. This is regarding to the reference. Thank
So
you.
I'm here to present one more paper of mine, that is fluoride removal from aqueous solution using granular natural soil and activated charcoal. So these are the contents of my presentation. So coming to the introduction, fluoride, it exists in the environment through both natural and anthropogenic sources. And also we know that it's essential for the humans, but when it exceeds certain range, it becomes detrimental. And it has been estimated that around 260 million people around the world in 30 countries are affected by consuming fluorinated water above 1 ppm. And in India alone, endemic fluorosis is thought to affect around 1 million people and is a major problem in 17 of the 29 states. And especially it can be significantly observed in Rajasthan, Andhra Pradesh, Tamil Nadu, Gujarat, and Uttar Pradesh. And approximately 62 million people, including 6 million children, suffer from fluorosis in the country due to consumption of fluoride contaminated water. So these are few of the ill effects of fluoride content. If it is less than 1.5 ppm, then it is going to affect the um, like bone development or skeletal development in children, and sometimes it leads to dental caries. And fluoride, when it exceeds 1.5 ppm, then it leads to tooth issues, skeletal fluorosis, and also bone crippling is being significantly seen in the humans. And in this particular study, from the literature, uh, the absorbents that were being selected are the natural soil, and the activated charcoal was being used for the fluoride removal. And an attempt was being made to check the effectiveness of the thermally treated soil adsorbents along with the activated charcoal. So the main objectives of the study include to study the selective locally available natural adsorbents such as the red soil, black earthen soil, and activated charcoal for fluoride reduction in water, and to evaluate the effectiveness of granular adsorbents prepared by thermally treating the clay balls made out of red soil, black cotton soil, and activated charcoal. The adsorption studies were also being carried out to optimize the adsorbent dose and also to understand the effect of contact time and the effect of pH. And the absorption study was being modeled using isotherm models, and the kinetic study was also being done. And the surface characteristics were also being studied for the adsorbents by using SEM and EDAX methods. So the methodology, it includes collection of the materials, and then the second one is preparation of adsorbent granules. Here, how it was being done is red soil and black cotton soil. They were being sieved through 75 micron sieve, and finally they were being completely washed, and the washed water passing through the 75 micron sieve was being collected in a container, and it was being sedimented there, and the clear supernatant was being taken out, and the sediment, which is the finer clay particle which settled at the bottom, they were being dried for 24 hours in an oven for 100 degrees centigrade. And then after that, the activated charcoal was being taken in appropriate ratio. I'll express the ratio in the next slides. And then it was being mixed, and they were being molded, that is the balls were being prepared, and then finally it was being baked using the muffle furnace at 1000 degrees centigrade, and they were being used as the adsorbents. And the absorption study, it was been done to find out the optimum adsorbent dose, and also the effect of contact time was been checked, and the effect of pH by carrying out the adsorption with varying pH values. And adsorption isotherm modeling for Friendlich and Langmuir were being done, and the kinetic study for the pseudo first order and second order was being done, and the characterization studies were being done by using SEM and EDAC. So this is the procedure that I was talking about. That is, the sieving and washing, etc., was being done, and finally the clay, black cotton soil, red soil, and activated charcoal powder were being mixed in a ratio of 1:1:0.5, and then granules of approximately 5 mm in diameter were being prepared by mixing all these ingredients using distilled water, and then the granules so prepared, they were being dried in the oven, and then they were being baked in the muffle furnace at 1000 degrees centigrade for 2 hours. So you can see here in the crucible that they are red hot, and finally they were being cooled in a desiccator, and they were brought to room temperature, and further the granules were being characterized before adsorption, and they were also being used for adsorption, and they were being characterized. So the absorption studies, as I told, it was being done to optimize the dose and also to study the effect of contact time and pH. So when the in the optimization study, it was found that the adsorbent dose of 4.8 grams was found to be optimum dose in reducing fluoride in the water from 5 milligram per liter to 1.46 milligram per liter. And from the study that was being done to study the effect of contact time, it was observed that 60 minutes was optimum contact time required for reducing the fluoride by 70.8%. And to when it was being tested for the effect of pH, it was being significantly observed that the fluoride removal was significant in the pH range of 6 to 10, but whereas the highest reduction was observed for the water with a pH value of 8. And the isotherm models which were being studied, it was a best fit for Langmuir isotherm model with a R-square value of 98%, which exhibited that it was the monolayer adsorption that happened there. And the pseudo second order kinetics model was the best fit as far as the kinetic study was concerned. From the SEM images, it was significantly seen that there was the textural change being observed in the adsorbents. A is the image of the adsorbent before the adsorption was being done, and B is B is the image of the adsorbent after adsorption. And these are the EDAX results, wherein significantly it was seen that after the adsorption study was being done in the D picture, here we can see that there was presence of sodium fluoride being observed, that is due to the adsorption over the adsorbent surface. So finally, to conclude, the thermally treated granular medium prepared using combination of red soil, black cotton soil, and activated charcoal of 4.8 gram showed a fluoride reduction of 70.2% for a contact time of 1 hour with an initial concentration of fluoride of 5 milligram per liter. And absorption of fluoride was significantly observed in a pH range of 6 to 8, which is well within the pH range of drinking water with, as suggested by Bureau of Indian Standard, which is in the range of 6.5 to 8.5. And the isotherm model study gave better fit for Langmuir isotherm with the R-square value of 0.98, which signifies monolayer adsorption. And from kinetic study, it was observed that the adsorption followed pseudo second order kinetics, and the surface morphology of the granular medium using some reflected textural change due to adsorption of sodium fluoride onto its surface. And also the EDAX results showed the addition of sodium fluoride in the composition of the adsorbent after the adsorption study. So this study, it indicated that thermally treated, locally available black cotton soil, red soil, and activated charcoal in combination in the granular form can be effectively used for defluoridation of water. So these are the references which were being used for the study.
Okay, sir. Good
Yes,
afternoon to
yes.
all of you present here. I am Anand Kishore,
Yes,
Assistant
yes.
Professor in Department of Food Engineering, Niftam Kundli. So I am presenting today the development and characterization of citric acid and heat moisture modified starch-based biodegradable film for food packaging. So as we know, the packaging is one of the integral part of food processing industry because it helps to improve the quality and shelf life of the food product. Generally, we say food is life, but if you relate the packaging, packaging is a life for food. So this is very important to understand how interact the packaging material with the food so that we can improve the shelf life of the food product. If you see the journey of the food packaging, it started from the leaves and woods and animal skins, and it's then the metals and plastic, glass. And nowadays, that all this form of the packaging material is converted into the plastic, right? So if you see all type of food product is nowadays packed in the plastic in the different format. Like generally in the food packaging, we use the LDP, SDP, high density polyethylene, polystyrene, polypropylene in the different format. So, but because why we are using the plastic in all type of food packaging material, because it has very good barrier properties, mechanical strength, that it has very good transparency, has very sealing properties at the lower cost. So this is the reason. But however, there is some problem with the plastic related to the environment as well as the health concern. So there is a migration and non-biodegradable nature of the plastic affect the environmental concern and the safety of the food product. So recently, the industry regulatory bodies and consumer have preference toward the bio-based packaging system with improved properties. So you can see the plastic in different format we can use. So this is the uniqueness of the plastic. It is versatile, it is flexible, we can convert. So the problem is the degradation is the major problem. So it degrades many of the year. So it affects the environment. So it affects the environment and the threat to the biodiversity. So also it affects the environment as well as it affects of the health effect of the consumers, because there is some chances of the migration of the microplastic from plastic bottles to the food materials, the plastic containers, the plastic tea bags, and there is some release. So in long term, it affects the health of the human. So what is the solution? The solution is the sustainable packaging system, right? So sustainable packaging is a holistic concept that develop packaging material to remain safe throughout their life cycle and meet packaging function at optimum cost. So many research are going on to develop this eco-friendly and bio-based packaging material from a starch-based polymer, cellulose-based PLA, PHA, and BioPET and BioPE. So our research group is published a review paper, and we found that the starch-based polymer is the most popular form of to develop the biopolymer. So, we and the main conventional starch we are using like potato, rice, corn, cassava, like this. So what we found that it has wide range of industrial application, like in the food formulation, pharmaceutical, paper coating, film forming, and packaging and textiles. But the problem is it competed with the food, because the conventional starch like potato, cassava, we are in the industrial application, but it affects the food requirement of the increasing population. So therefore, worldwide research has recently focused on extracting a starch from underutilized crop like root, tuber, seeds, so that we cannot compete with the food. So I select jicama starch for my work for development of the biopolymer. So jicama is one of the non-conventional stores of the starch, belongs to the family of leguminous and cheap source of carbohydrate with good nutrition value food. And it has significant amount of starch which is replace the conventional starch. So recently we also reviewed this in a National Journal of Biological Micromolecules, and we found that there is some, that means very less research on this to modification and application in the biopolymer side. And also after review, we found that there is a challenges for the starch film. It has less mechanical strength, as well as it has very bad barrier properties in terms of water vapor transmission rate, oxygen transmission rate. Its sealing is not good. So there are different strategies have been adopted by the researchers to improve the starch-based biopolymer. right? So by the different technique like modification, reinforcing the fillers, cellulose, the composite material, we use starch and protein and fiber, and we develop the multi-layer films, adding some active material to the starch so that we can make it active and we can compete with the your synthetic polymers, right? So in this study, I use the modification technique in which I just modify the starch. Just I work with the raw material. We start modify the starch with the HMT, high heat moisture and treatment, and citric acid, and we compare the effect of this HMT and Ca with the native starch in terms of their starch characteristics, as well as we develop the film from that, and we compare the film. So my method is divided into three phases. The first phase, we just extract the starch by grinding, cutting, peeling, and this sedimentation technique. And after that, we go for the characterization. In first, second, we just that is the native starch. And the second phase, we just modify the starch with HMT. So HMT is the physical modification technique where the starch granules is heated at very high temperatures. So here I used 150 degrees centigrade, 120 degrees centigrade with a moisture content of 20%, and the time for heating is half an hour. And again, citric acid, we use the 20% of the weight of the starch. So after getting this modified starch, we go for the characterization of the starch. And after that, we just make the film from solvent casting method from native and the modified starch. And we did the characterization and we understand what is the effect of this modification on the film quality. So first is the effect of treatment of your physical property of the starch. The first, the amylose and this swelling power and solubility and the color parameters. So amylose is very important, the crucial role for the starch properties because it affects the film forming ability. So after modification, we found that HMT has higher this amylose content. Because after the heat treatment, maybe the chances of this breakage of the amylopectin and the bleaching of the amylose, so this value is very high. But in the case of citric acid, the value is less if you compare with the native starch, because if we apply the citric acid, so we have the cross-linking. So it strengthens the amylose, so it reduces the leakage of this amylose through that structure. So it may be the reason behind that there is a reduction in the amylose content. Swelling power and solubility is again the most important parameter of the starch to understand the hydrophilicity and the hydrophobicity behavior of the starch. So this is very important for making the film, because the starch-based film has the problem with the hydrophilicity. So the purpose of modification is that how we can make it hydrophobic starch. So by the modification, we can see here there is some reduction in this swelling power and solubility after this treatment. The color parameter also there is some lightness, that means the transparency, that lightness is increases. So it affects the color parameter also. So if you can see the effect of treatment of the thermal property of the starch, so thermal properties, you can see the transition temperature increases by after modification of Ca and this your HMT. So the gelatinization temperature of jicama starch increased basically with HMT and Ca modifications that indicate the stable configuration and better thermal stability if we go for the further processing. As well as if we see the gelatinization enthalpy that decreases in all modified starch. However, in the citric acid is more as compared to the HMT starch. It may be the reason that citric acid do the cross-linking, it strengthen the amylose and amylopectin arrangements. So that may be the reason. Next is again the effect of treatment, the pasting property of the starch, the peak viscosity, breakdown, final viscosity, and the settler viscosity decreases after the modification. But in the pasting temperature, if you see the after modification, the value is increases. So the pasting temperature increases. That means this is very good thermal tolerability after this thermal processing. But in the case of HMT, the value is high if you compare with the citric acid. So it may be that due to the heat treatment, there is some rearrangement of the amylose and amylopectin. So we have this stable structure. That's why it may be the reason that the pasting temperature is high if in the HMT treatment. In the case of, we did some XRD patterns. So we found that there is no any, that means change in the pattern. So we found the A pattern, the major peak is 15.4, 17.2, and 18.2 and 23.1. But there is some changes in the crystallinity patterns. So if we modify the stars, there is a decrease in the crystallinity. The native stars has 35% and this 34 and then 33. So it may be the chances of citric acid, again, we have the cross-linking, it affects the crystallinity. And in the case of HMT, during the high heating treatment, they just destroy the crystallinity we have here. So maybe the chances of to reduce the crystallinity of the that HMT starch. This is the molecular structure based on their functional group. We found that there is no any major difference, but we found that one peak is 1706. The formation of new functional group in case of CA modified starch, maybe due to the citric acid cross linking. The big bond was absorbed around 1148 for all type of samples, and major peak we found that 1008 centimeter inverse. If we did the same analysis, that effect of treatment, the morphology of the starch, we found that there is some cluster formation on this after modification, the CA and the HMP starch. So the cluster formation, maybe the found that after the modification technique, there is a formation of bond between the molecules. So that may be the reason there is a formation of the cluster, and the surface is not, that means packed structure if you compare it with the native starch. So after that, we did some that we developed the film from this modified starch and the native starch, and we compared the characteristics, and we found that the effect of treated starch on physical properties of the film, like thickness, moisture, water solubility, the color, and the opacity. So thickness, there is no any significant difference, but thickness is very important parameter to check the quality of the film, because it affects your tensile strength as well as the variable properties. But in this case, there is no any significant difference in the thickness. It may be the reason that thickness basically depends on these formulations and the that means your volume of your samples. But in the case, in all this sample formulation is same, and the volume we have taken the same. So maybe the reason that the thickness, there is no any significant change in thickness, but moisture and water solubility has again the lower if we modify the starch. Similarly, the affects the color parameter also. The lightness value is increases in the case of the modification. The opacity is also increases in the case of modification. So it better sign to, that means it affects the modification technique on the quality of the film. In case of water vapor permeability coefficient and oxygen permeability coefficient, this is very important parameter which affects the shelf life of the packaging film. So after modification, there is a decrease in the water vapor permeability coefficient. Maybe the chances of because we modify the citric acid, we have the cross linking, and so we found in the water solubility and swelling power of the starch, it reduced the, that means hydrophilic characteristics. So maybe the reason that after modification there is a reduction in water vapor coefficient. Now oxygen permeability coefficient also the important parameter. So again there is a decrease, because this affects that means after modification there is a rearrangement of the amylose and amylopectin. And also we see the same structure, there is a uniform and cluster formation. So they make the bond, a strong bond between the molecules. So the diffusion path is reduced actually in the case. That's why the WBPC and OPC is reduced. In case of tensile strength, increases after modification. But in the elongation and break, you can see after heat moisture treatment, the elongation and break is increases. But in the case of citric acid, it is decreases. So in the packaging film, actually the tensile strength should be high and elongation at break should be high. But in the case of citric acid, the value is less. So it may be the chance that we develop the cross-linking, so it reduce the mobility of the molecules in the structure of the stars. That's why there's a reducing the elongation and break if we compare with the HMDT. Next, same analysis we found, and we found that the surface of the film is very rough in the case of native starch, but in the case of citric acid and this HMT treatment, the surface is very homogeneous and the structure is regular. So it may be the chances it affects the properties of the film, and there is an improvement, we can say, in the properties of the film. So the conclusion from my research project is the modification of native starch change the properties of jicama starch, which can be used in fine suitability in the food formulation as well as the packaging industry. The film prepared with HMT and CD modified starch showed promising result with increase in tensile strength and improve the film barrier properties. HMT modified starch-based film barrier superior if we compare with the citric acid in terms of the tensile strength, water vapor permeability coefficient, oxygen permeability coefficient, elongation and break, as well as the opacity. So the finding of the study show that modifying the jicama starch with HMT and CA treatments had a significant impact on starch properties and improve the film quality for the further applications. However, there is a need, if we compare with the synthetic polymer, so there is need to work on this direction so that we can complete the properties with the synthetic polymers. These are my differences. Thank you.